All right, let's go ahead and get started. So thanks everyone for being here for Department of Medicine Grand Rounds. We are very excited to welcome Dr. Douglas Walker, who is an associate professor in the Gangarosa Department of Environmental Health here at Emory. He's an environmental engineer and analytical chemist with training in metabolomics and developing exposome-wide association study methodologies for environmental health and precision medicine research. His research focuses on continued development and application of advanced analytical strategies for measuring the occurrence, distribution, and magnitude of previously unidentified environmental exposures and assisting in delineating the mechanisms underlying environment-related disease in humans. The approaches he developed showed it's possible to measure over 100,000 chemical signals that include exposure biomarkers, nutrients, dietary chemicals, and associated biological response in a high throughput and cost-effective manner, establishing a foundation for operationalizing the exposome framework for precision medicine. Ongoing research projects are now focused on using high-throughput exposome methods to establish disease exposome atlases and development of methods for measuring biomarkers of complex exposures of emerging concern, including microplastics, e-waste, and polyfluorinated chemicals. He leads the Comprehensive Laboratory for Untargeted Exposome Science, which was established to provide high-quality, untargeted screening of biological samples for nutrition, precision medicine, and environmental health research. Thank you for being here, Dr. Walker.
Great. Yeah, I'm very excited to be here. I actually spent 5 years, I guess, technically in the Department of Medicine, although I was in the Division of Pulmonary. So it's really great to be back at Emory and presenting on the work we've been doing and kind of highlighting the capabilities that we have in our lab. So as you heard, my name is Doug Walker. I'm an associate professor in the Gangarosa Department of Environmental Health. Our laboratory is actually located right on the Emory campus. This is the Claudia Nance Rollins building right here. Our lab is in some of these windows. So if you're interested in kind of seeing more of what we're doing or learning more about these techniques, you're more than welcome to shoot us an email and stop by and kind of learn a bit more about our research. So before I get started and really start talking about the different methods we're using, I really want to acknowledge our team that has contributed to development of these microplastics methods. These have been in development for over 5 years at this point. The measurement of microplastics in biological samples has been an extremely difficult undertaking, and we have learned a lot along the way. And I really want to acknowledge the team members we had first at Mount Sinai and now here at Emory that have really contributed to advancing these methods and really bringing them to where they are today. I'm not just saying this to make myself sound important, but I really am convinced that there's only a few labs, in the entire world that are capable of measuring these microplastics. And, thanks to all of the hard work by this team, I truly believe that we are one of them. So I'm very proud of all they've accomplished, and it's been, a delight to work with all of them as they really, are the ones responsible for advancing these methods. So, just to start, I wanted to give a very brief overview and perspective on how we approach this question of addressing environmental factors and human disease. And so, our current estimates suggest that only about 5 to 15% of diseases can be attributed to heritability or your genetics alone. The other 85 to 95% of diseases are really derived from the environment, lifestyle factors, and their interaction with the genome. So, this genetic or genomic perspective that we've taken to really try and understand what drives disease outcomes and disease diagnosis and disease risk is really giving us an unbalanced view of human health and disease. So, what our research does is really try to provide more insight and information on those environmental factors and how they may be linked to different disease outcomes. And with all things, especially in research, this is much easier said than done. So our current estimates suggest that there are over 350,000 chemicals that have been registered for production and use worldwide, of which over 50,000 remain unidentified due to confidential business information. And so when we start thinking about what our exposures are as humans and what are the things that we come into contact with that may impact our health, the challenge to actually measure and understand these is immense. Not only is there a huge quantity of exposures that we need to consider, but there's also, many that we may not even be aware of or may not even know are ongoing exposures in human populations. So, this need to better understand the environmental factors that drive health and to kind of complement our studies of the genome with a way of including environmental factors led Christopher Wilde to introduce the concept of the exposome back in 2005. And this has very much been a growing area of research, one that Emory has actually been at the forefront of, thanks to uh, faculty like Gary Miller and Tom Ziegler, who's on here, and Dean Jones, and many others. And as this area or this research area has kind of grown and evolved, we've kind of viewed this concept of the exposome as the integrated compilation of all physical, chemical, biological, and psychosocial influences that impact biology. And what we're attempting to do with this exposome concept is essentially develop a construct very similar to the genome, where we're trying to systematically evaluate the different factors that may contribute to disease risk. And when we talk about the exposome, it's actually a very broad research area and kind of topic because that essentially includes all non-genetic disease risk factors. So this can include things like ecosystem or lifestyle stressors. It can include social stressors, as well as physical chemical exposures, which may be from pollution or heat or noise or many other types of exposures. And so our research really focuses on this concept of addressing the chemical exposome. While it's very important to consider how the chemical exposures interact with infectious disease exposures as well as stress exposures, our specific research focus in my lab is developing methods that allow us to better understand the chemical exposome. And we primarily do that through developing analytical methods that allow us to measure biomarkers of exposure in biological samples. And this can be things like blood or urine or tissue. And we try to use those exposure profiles to estimate what somebody's exposure history is and how that may be associated with future risk of disease. However, there's a discrepancy between our ability to actually measure environment compared to some of the other techniques we have for very comprehensive characterization of biology. So, over the past 20 years, we've seen tremendous advances in approaches that allow us to measure different aspects of our biology. We've seen the price of whole genome sequencing and genotyping drop significantly. You can get a whole genome sequence for about $700 now. You can spit in a tube and send it to whatever 23andMe becomes and get your genotype back for $100 or whatever. But at the same time, we've seen these huge advances in next generation sequencing that tells us a lot about gene expression. We've seen changes in our ability to measure the epigenome, as well as very high throughput proteomics methods such as SomaScan or SomaLogic and Olink. But many of the capabilities that we're using to study environmental exposures are still using approaches that only allow us to characterize about 10 to 100 exposures at a time. So, what my laboratory focuses on is this critical need to develop analytical frameworks that provide us more universal screening of exposures so we can use a data-driven approach to identify which exposures are actually associated with increased risk of future disease. And we do that using a number of different approaches, but kind of the primary strategy that we rely upon on our laboratory is the use of a platform called high resolution mass spectrometry. And what this system allows us to do is it can both detect very low levels of biomarkers in a complex sample, and it doesn't require us to a priori identify what are the signals we're going to be measuring. So we can actually use these instruments to detect molecules that are present in a sample, as well as predict what they might be or try and predict the identity of those compounds. And the reason we're able to do that is the high resolution mass spectrometers allow us to detect a mass in our instrument to a very high degree of accuracy. And we can actually use that mass to try and predict what the chemical formula is for that compound. And while there's a lot of nuance and different approaches for going from that signal to annotation, it can still give us hints about what are the things we're measuring and doesn't require us to necessarily know the exposures we're studying or measuring that may be linked with our outcome. So very similar to how we do like a genome-wide association study, these instruments are allowing us to perform essentially an exposome-wide association study where we can systematically test the different biomarker measures that we have from our instrument to see which are linked with a disease or outcome or and may be contributing to increased risk for that. So what my laboratory is really focused on is using these high resolution mass spectrometry instruments to kind of link three critical areas in really understanding how environment drives different health outcomes. So our first focus is leveraging high throughput and comprehensive exposome methods to really try and identify both exposures associated with future risk of an outcome, as well as early biological changes that may explain some of the mechanisms that that explain that exposure disease association. We're also developing these exposomic methods that can be incorporated within different bioeffect or multi-omic studies. And this allows us to essentially prioritize what are the key exposures that we're measuring that may be linked to a disease process or a disease mechanism. And at the same time, we're also trying to develop new methods for characterizing complex exposures of emerging concern. So these are things that we may recognize are important exposures in human populations, but for a number of reasons, we may not have good biomarkers for them or good ways to measure them. And so my lab really uses this advanced technology base that we have to try and advance these newer methods for studying those exposures in human populations. And so in terms of our more routine workflows and kind of some of the capabilities we have in the lab, our goal has really been to try and create an open and fair exposome analytical framework. And this is more for general study of exposure disease relationships. I'll go more into the microplastics following this. But we've essentially developed robust and large-scale methods that allow us to characterize the exposome in tens of thousands of samples. We have almost completely automated sample preparation methods. We have high throughput analytical approaches, as well as very comprehensive platforms for characterizing the exposome in different types of biological samples. We've tried to do this in a way that improves accessibility for environmental health and biomedical researchers who may not be analytical chemists or mass spectrometrists, but we're still trying to generate data that can be used and incorporated into, exciting and new areas of research where maybe there hasn't been as much emphasis on environment, prior to that. We're also trying to do this in a way that allows us to establish cumulative exposome disease atlases. So a lot of the research that's been done to date kind of views individual studies in isolation. So for example, I may have a study of diabetes where I have 500 samples from my cases and controls. If I was analyzing those samples, I would generate all the data, extract it together, analyze that data for whatever I'm interested in for outcomes, and then write up my studies or write up my papers. And that's kind of it. And then maybe after that, I'll move on to another study of cancer or Alzheimer's disease. What we're trying to do is build a framework that will actually allow us to integrate all of these diverse studies. So we can look at exposure profiles and associations across both similar and different disease processes to kind of see what are the key exposures driving, human health. And finally, we're also trying to do this in a way that allows us to integrate this exposome data into multi-omic approaches so we can, again, kind of prioritize what are the key exposures that may be impacting pathways or biological systems. And so, just as kind of a summary of what we have in the lab, I've been here at Emory for about 2.5 years now. Before that, I spent 4 years as a faculty member at Mount Sinai. Before that, I was a PhD student and postdoc, kind of hanging around Emory and working in the pulmonary division. But since moving back to Emory, we've established a cutting-edge exposome research laboratory really developed for high throughput exposomic research. We have capabilities that allow us to measure the exposome, metabolome, lipidome, and many different kind of small molecule profiling methods. Our technology base has vastly grown in the past 2.5 years. We now are operating two gas chromatography high resolution mass spectrometry systems, which were right now will primarily be used for microplastics. We have four liquid chromatography high resolution mass spectrometry systems, as well as a targeted gas chromatography system that will be used for very low level biomonitoring of different exposures. And we also have, very, comprehensive facilities to minimize contamination during sample preparation, which is very, very important for these types of studies, especially the microplastic studies. So if you're interested in collaborating or kind of learning more about these capabilities, please feel free to reach out. So, after that kind of quick description and overview of what we're doing and kind of the philosophy we take to incorporating these measurements into studies of human health, I'll now like to move on to how we're leveraging these analytical approaches developed for the exposome to provide new measures of microplastic exposures in human populations so we can really understand how, what exposures are occurring and how they may be impacting populations. So my presentation is going to focus on kind of three main topics. The first is really how we can move beyond simple measures of plastics to really characterize what is actually a very, very complex exposure. I'll next describe some of the analytical challenges and opportunities in microplastics research, specifically how we're applying high-resolution mass spectrometry for scalable assessment of plastics exposure. And then I'll spend a little bit of time talking about some of our current studies where we're actually using these methods to measure microplastics in different types of tissues. So before going forward, I think it's very important to just, take a second or a few minutes to kind of remind us all of what plastics are, how they're used, what are the different types of plastics in use, and kind of what happens to these plastics when we're finished using them. So plastics themselves are essentially long, very, very long molecules that are built from individual building blocks called monomers. And these monomers are essentially small molecules that are refined or collected during oil and natural gas production. So these are actually a byproduct of oil and natural gas production. In order to create these plastics, these monomers undergo a polymerization reaction where the individual monomers essentially link to each other and then form these very, very long molecules called polymers, which can have molecular weights of, tens of thousands of Dalton. These polymers are then formed into plastic pellets, where they can be formed in, or they can be used for manufacturing a number of different products. However, plastics themselves, or the pure mono or polymers themselves, often don't have all of the properties that we would want for our plastic products. For example, some of them can be broken down by UV light very easily. If it's just that pure polymer, they can be very brittle and may not have the properties that we want for the products that we're trying to create. So often plastics are not just the polymers formed together, but they also contain a number of different additives that change the properties of that plastic so they can be used for their ultimate applications. And you may have heard of some of these additives, but these include things like plasticizers such as phthalates or bisphenol A, flame retardants, stabilizers, antioxidants, colorants, and many, many other additives that are included in these plastics to, again, kind of make them more usable for the end application. So I don't think it will be a surprise to anybody, but we've seen a tremendous growth in global plastics production since 1950. These numbers are a little out of date, so this only goes up to about 2015. But cumulatively, we've seen almost 10 billion metric tons of plastic materials produced since the 1950s. And this rate has actually increased significantly since COVID. The annual plastic production has increased. As of 2015, it was about 300 million metric tons of plastics that are being produced. And again, that number has grown even more over the past few years. And the use and formulations of these plastics are extremely diverse, and the number of plastics or the amount of plastics that is produced varies significantly by type. So the most commonly produced plastics include things like polyethylene, polypropylene, and PVC. But there's also a number of different polymers and copolymers that are being produced. And as you can see, many of these are being produced in the millions of tons each year. And the application of these plastics and the amount produced for these different applications also varies significantly. So packaging is by far the largest use of plastics, particularly single-use plastics. But these materials are often also used in things like building and construction and textiles and transportation and many different applications. And so, if you're ever interested in what are some of the plastic materials or plastic formulations that you're using, you can actually tell that by the recycling number that's on the bottom of your bottles or plastic materials. So I'm sure you've all seen the recycling symbol, and it has a different number in it. And that number is actually telling you what type of plastic it is. And that kind of helps guide how it should be recycled or what should it be recycled with. So for example, you can see number two is high density polyethylene. Number three is polyvinyl chloride, which is used in drinking water pipes. But again, this can kind of tell you what the different type of plastic is. And of course, their actual use and formulation is very, very different. And they also have very different properties. So I think one thing that has really been shocking to me as I've moved into this plastic field is really just how frequently or how often plastics are just used once and then discarded. The amount of plastics that are actually being recycled is only about one third, or not, I guess not single use is about one third. And the amount that's actually recycled is only about 10% of the total amount of plastic that's been produced. The majority of it has just been single-use plastics that once it's thrown away, the majority of it will go to a landfill or it's incinerated to basically destroy it. And I won't kind of go into this in more detail, but if you're interested, this kind of shows you just the distribution of different plastics, what their applications are, and kind of what happens to them after they're used and discarded. And so what really we're interested in now and what our research focuses on is, essentially what happens to plastics during their use and after they are discarded. And so plastics themselves can actually undergo a number of different weathering processes that will either release additives that are present in the plastics or can cause shedding of plastic particles from these materials. These processes are very diverse. Some microplastic particles can be created through weathering in landfills or in the ocean. Other sources of microplastics can be through cooking food and plastic containers. But basically, there's many different sources of microplastics, and these can, just based upon the volume of plastic that's being produced, these have the potential to result in a huge ecological and health burden. When we think about microplastics and their formation, there's kind of two different routes of production. The first is primary microplastics, and these are essentially materials that were manufactured as small plastic particles. And then the second category are secondary microplastics. And these are actually from the weathering and degradation of plastic materials. And you can see if you're considering sources of microplastics, some of those common sources are things like personal care products, tires. Your tires actually release a little bit of rubber every time you're driving on them. Textiles, I'm sure many of you have emptied the lint filter in a dryer. Those are microplastics. But really, the largest source is weathering of these macroplastic products. And these are essentially larger plastic products that, as they degrade or as they break down, they form into smaller microplastic particles. And so it's very important to start thinking about size when we're actually talking about these microplastic exposures. And so microplastics and nanoplastics have been measured over a very large range of size, from things that you can actually pick up at the beach and see with your own eye, through things that are smaller than cells, smaller than viruses, and even down into the nano range. So just to give you a little bit of a context, the particles or the microplastics that we're most interested in studying for human exposures are essentially particles that are about 5 micron or smaller. And so for context, a 10 micron microplastic particle, you could essentially stack five of those together, and those would be the width of one human hair. Particles that are smaller than 2.5 microns, you could essentially fit four of those in that 10 micron particle that we stacked across a piece of hair. So you can tell these are very small particles. And in fact, from an exposure perspective and what we can measure in humans, really we're interested in things that are smaller than 1 to 5 micron. And so, just for some context, a particle of smoke may be about 100 to 700 microns. The SARS-CoV-2 viron is about 60 to 140 nanometers. The width of a strand of DNA is about 2 nanometers. And the measurements we're using or applying are allowing us to measure microplastics from confirmed about 300 nanometers and larger, but we may think that due to some aggregation or clumping while we're manipulating our samples, we may be able to measure, even down to 100 nanometers or smaller. And I'll talk about that more in a few slides. And so I'm sure many of you are aware or have heard of the microplastic studies that have been coming out. There's been a lot of press on these lately. Some of it deserved, maybe some not. But it seems like just about every other day, there's newer papers coming out showing that microplastics have been measured in some tissue or some biological sample. And so this is from a review that was published in 2024, just showing all of the different types of biological specimens that have had microplastics measured in them. And it ranges from everything from, blood, breast milk, your lung, placenta, and so on. But something that isn't really appreciated in the current approach to microplastic research is really just how complex these particles are and how important it is to consider this complexity when we're trying to understand how microplastic exposures may impact human health. So, for example, microplastics can include many different sizes and shapes, from fibers through particles through shards. They come in very different sizes, ranging all the way from, nano-sized through micro-sized. They can include many adsorbed or associated pollutants. So, there's been some evidence showing that microplastics can form biofilms and where basically bacteria will form around these particles. There's also evidence suggesting that other environmental pollutants can actually stick to the microplastics and be transported with them. At the same time, like I mentioned, microplastics or plastics aren't just the polymer alone. They include a lot of different additives and materials that have been included within the plastics, again, to make them more suitable for whatever the end application is. And so it's really important to start considering this complexity when we want to study the health impacts of microplastics. And this has been even further highlighted by the PlasChem report, which came out early last year, which basically tried to do an accounting of the different chemicals that are used in plastic manufacturing and provide some insight into what we know about those different chemicals and additives. And so, what they were able to find from this report is that over 16,000 different chemicals are used in the process of plastic manufacturing. And many of these, we don't have any data on the potential health impacts, the information on release of these chemicals, and whether these are things that we should be concerned of from a human exposure perspective. And this is just highlighting that further. So, what this is showing is the categorization of different chemicals that are used in the manufacturing of different plastics based upon whether toxicity information is available for these chemicals. And so if you see something that is red, it suggests that there is some evidence suggesting that these chemicals could potentially be hazardous or toxic if there are ongoing exposures to them. The orange suggests that there may be some toxicity associated with it, but it's not, very strong evidence. The green says it's not hazardous, and then the blue is just the compounds that have no information at all. There's been no assessment of the toxicological relevance of those compounds. And you can see from the plot on the right, which has essentially converted these raw numbers into proportions, for many of the different chemicals used for the manufacturing of these different plastics, over half of them have almost no information on their toxicity or hazards. And at the same time, there's a huge amount of these materials that are being produced. And what this is showing is the production volumes for differing numbers of chemicals that are used in plastic manufacturing. So you can see that for about 4,000 chemicals, there's greater than 1,000 tons produced and less than 10,000 tons of it. So that's a huge amount of materials. And for many of them, up to 500 of these chemicals, there's over 1 million tons that are being produced each year. So not only is there the potential for exposure to a wide range of chemicals, but this isn't a very rare material that may be manufactured in a lab or it's a very low production volume. This is some of the highest production volume chemicals that are currently being used in the world. And unfortunately, our current biomonitoring approaches, and biomonitoring is essentially using different exposure biomarkers that can be measured in a biological sample to try and assess exposure to different chemicals. Our current biomonitoring approaches do not account for this complexity of plastics-related chemical exposures. Currently, our methods that are in use only allow characterization of about 10 to 20 plastic exposure biomarkers. These include things like phthalates and bisphenols and different manufacturing additives, and it's largely ignoring many of these other chemicals and other compounds that are used in plastics. For example, the micro and nanoplastic exposures aren't being characterized. There's very little information on these additive and processing aids and what these actually may be transformed into. And so what we're trying to do is really, embrace this complexity and develop new methods that allow us to better cover plastics exposures, in particularly to micro and nanoplastics. Unfortunately, there isn't any gold standard method to measure microplastics. And there's also many different aspects of microplastics that are important to measure. And so the kind of primary approaches that have been used in the past have kind of can be classified into two different flavors. The first is particle-based, and these are methods that essentially count particles and measure their morphology or their size. And this is based upon the use of different spectromicroscopy methods like FTIR or Raman, or even for the smaller particles using microscopy methods like scanning electron microscopy or tunneling electron microscopy. The other strategy, which is what we focus on, is using mass-based approaches. So these techniques don't necessarily allow us to characterize the number of particles or their size, but it does allow us to assess an exposure burden so we can see what that's associated from a health outcome perspective. But I do just want to highlight that, these exposure assessments are very complex, and there's really no single analytical method that can provide complete information on micro and nanoplastics. And that's very important when we're interpreting our studies. So the approaches we use are based upon body burden and measuring the levels in tissue or samples. There's a lot of effort going into these particle-based methods, which look at morphology and composition. There's development of different methods that actually allow you to look at compartmentalization. So microplastics are particles, they're not molecules. And it's unclear about whether these may accumulate in specific interfaces or tissues relative to others. And of course, we're also very interested in studying co-exposures and other things kind of transported on these pollutants. And unfortunately, even though probably most of the methods or the primary approach to measuring microplastics has been microscopy methods, they're really not suitable for measuring microplastics in biological samples. And so when you're using microscopy methods, you need to use some type of approach to actually verify that the particle you're imaging is actually a plastic. And so the most common approaches that have been used are things like, FTIR or Raman spectroscopy, which allow you to both image a particle and look at the spectra from it to identify what the underlying polymer is. Unfortunately, these often are limited by resolution and do not allow us to measure particles that are smaller than 1 to 5 micron. And so it's really not capturing that nanoplastic range. Of course, we can use things like electron microscopy or scanning probe microscopy to do that, but that only allows us to visualize the particles. It doesn't give us any chemical composition information. So we can't actually confirm the particle we're seeing is a plastic. And now, this kind of is in opposition to the biological plausibility of what we will be measuring from a microplastics perspective in different tissues. And so, Many of our microplastic exposures are coming through the food we eat, and so that would require that they cross our gut barrier. And really, our current estimates suggest that, particles less than 2.5 microns are the only ones that can actually cross this barrier and be taken up into systemic circulation. And so these particles are so small that they can't accurately be visualized and they need to be measured using different analytical approaches. And so the way we've addressed this is through leveraging untargeted exposomic methods that allow us to both screen and quantify a wide range of polymers and plastic-related chemicals on a scale consistent with their exposure potential. And so I'm running a little low on time, so I'm not going to go too into detail about what that means. But essentially, these untargeted methods allow us to go in and detect as many signals as possible and allow us to prioritize what we should be focusing on from a disease outcome perspective. This is different from a traditional analytical chemistry approach where we develop our methods to only measure a very small subset of the biomarkers or exposures that we're interested in. And so, through funding we received from the European Union in support of the Aurora project, which I'll talk about in the next few slides. Our focus over the past 5 years has been to essentially develop what we like to think of as scalable exposure approaches for analysis of micro and nanoplastics. So the sample preparation steps are very complex for analyzing these. All of you that have been in labs know that just about everything in the lab is made out of plastic, so the risk of contamination is huge. And so, what we've really focused on initially is how to develop these protocols that give us a higher throughput capability for analyzing microplastics in different tissues. And I mentioned quality assurance, quality control. This is a key aspect of everything we do related to microplastics. For this, precaution goes a very long way. And again, I won't go into detail about the different steps we use, but this should absolutely be a critical consideration and first step in anything that you're doing related to microplastics and plastics analysis, is making sure you have the suitable QA, QC, and evaluation procedures to ensure that what you're measuring in your samples isn't some artificial isn't artificially introduced from your sample preparation methods. And so, in terms of scaling these methods, we've focused on developing an analytical workflow that we refer to as fast MPA, or fast single tissue extraction for multiplexed plastics analysis. And so within this workflow, we essentially use three different analytical techniques, or soon to be three different analytical techniques, to very comprehensively characterize the presence of plastics in biological samples. So to do this, we essentially take a tissue or fluid sample. First, as I mentioned previously, we are an untargeted exposomics and metabolomics lab. So oftentimes we will take a sample aliquot or biopsy before the microplastics analysis and run that for small molecules related to different exposures and endogenous metabolites. But then after that, we take a tissue extract or a biopsy, and this can range from blood to urine to follicular fluid to placenta, and we'll digest it to break down the tissues, but essentially leave the microplastics in their current state. Once we've done that extraction or digestion, and that occurs in a microwave system, so it's allowed us to accelerate how quickly we can do that from 48 to 72 hours to 30 minutes. We then take that digest and filter it to allow all of the liquid and dissolved molecules to pass through the filter and trap all the microplastics that are remaining. And the filters we use vary. For tissue, we're using zero 0.7 or 700 nanometer micron filters. For biofluids, the goal is to use 0.3 or 300 nanometer fluids. And after this filtration, we actually take three separate punches of that filter, and then we have three different ways that we can characterize microplastics on them, or soon to be three. One of the methods is in development. The first approach, and what I'll kind of focus on in the last few minutes of the presentation, is a strategy called pyrolysis with gas chromatography high resolution mass spectrometry. The next approach is alkali assisted hydrolysis with liquid chromatography high resolution mass spectrometry. What that does is it takes the polymers that are present in those microplastics and depolymerizes them, releasing additives and allowing us to measure the individual monomers. And then the last approach that we have all the infrastructure for, but we haven't started method optimization for this yet, is using size exclusion chromatography interface to a high resolution mass spectrometry system. And this will actually allow direct quantification of known polymers. All these other methods we have are called indirect methods because we're converting the polymers into something else before we're measuring them. But I'd like to next focus on pyrolysis, which has been one of the most commonly used mass-based measurements for microplastics and is really emerging as one of the key technologies that we're using in our lab. So with pyrolysis analysis, what you're essentially doing is taking those extracted microplastics and you are introducing them into a furnace that is rapidly heating them to between 600 and 700 degrees. At that temperature, those polymers and microplastics will combust and form pyrolytic products that we can measure on our instrument that we know are indicative of certain polymers. And so, what essentially happens is when we're looking at a pyrogram, that's called, and that's the results from the pyrolysis analysis, we know that certain molecules are forming from that combustion reaction. And so we know that if we're detecting that molecule, that is a measure of that polymer, and it suggests that that polymer may be in our sample. And so the reason that this is such a reliable way and something that we've really focused on is it can be used for a number of different applications, whether we're measuring water or tissue. It's very sensitive, so it allows us to measure very low levels of polymers. And using the high resolution mass spectrometry instruments we have, it can give us very detailed insight into polymer composition and polymer mass, as well as the presence of other additives. However, it's important to recognize there are some limitations of these methods. It does not allow us to assess particle counts or shapes, so it may not be comparable to some of the microscopy methods. But most importantly, there are some organic material that, when it's pyrolyzed, can actually break down to the same pyrolytic products as polymers. And if you're not removing those background interferences, you can incorrectly measure those compounds as plastics. And I don't want to be negative about this recent study that came out, and I think this was it's important to advance the methods, but you can kind of see that effect occurring from the pyrolysis analysis in this recent publication from Nature Medicine. And so what they did in this study is they had used a pyrolysis-based measurement technique that they applied to different organ systems, and then also looked at the difference between brains from individuals who had Alzheimer's and healthy controls. Now, there were some other methodological issues related to control matching, but one thing you'll see here is that the individuals with dementia had brain microplastic levels that were, 2 to 5 times higher than any of the other population. And so when you see an effect this large, it suggests a few different things. One is that microplastics basically cause Alzheimer's disease, which we know is not the case because there was Alzheimer's disease before microplastics. It suggests that there might be some confounding or reverse causality that's occurring. But in this case, I'm pretty strongly convinced in that they were looking, people with Alzheimer's may have very different brain compositions, specifically lipid compositions. And I think what was happening is they were essentially measuring artifacts from biological changes due to the pathobiology of Alzheimer's itself. Um, so I'll kind of move quickly through the rest of this since we only have a few more minutes. But one of the ways we avoid this, and also to kind of again embrace that complexity of what we're measuring, is we actually integrate two separate measures using the pyrolysis system on our instruments. And this approach is called a double shot analysis because when we're characterizing our samples, we first heat them to about 300 degrees. And this will cause many of the additives or other environmental chemicals to basically be released from the plastic particles that are in the sample. However, it's not hot enough to combust the particles themselves. So after this thermal desorption step, we then pyrolyze the samples, which is when we heat them to 650 degrees, and that causes the breakdown of the polymers to the individual products. We're doing a lot of other work trying to see what we can measure in these samples, but I did want to highlight that we're developing libraries of over 30 polymers that we can screen for in our samples. So we know that, what the signatures to these polymers should look like, and we can pull that data out with a high degree of confidence if we measure them. But at the same time, we're also integrating quantitative workflows that allow us to measure up to 12 polymers and provide absolute concentrations of those polymers in tissues. And then very briefly, I just want to kind of highlight the work that we're doing for the Aurora project, which was funded by Horizon 2020 from the European Union. And so the goal of this study was really to try and understand the early life impacts of micro and nanoplastic exposures and see if there are any association of in utero exposure with either birth or developmental outcomes. And so there's a number of different work packages and components of this project, but I'm really only going to focus on the first two, which is largely trying to do essentially deep characterization of microplastics in maternal and fetal samples to kind of identify key characteristics of microplastics in biological samples. And what's been really interesting about this is we've ultimately found that a lot of these microscopy methods just do not provide the selectivity and sensitivity to measure the small particles in biological specimens. They either need to have fluorescent particles or some way of kind of tagging the particles so they can be imaged. But really, it's been very challenging to apply these microscopy methods. However, applications for scaling mass-based measurements have been very successful. And by the end of this project, we will have analyzed 800 placenta samples collected from pregnant women in Spain or Belgium and characterized, microplastics in these placentas, and we'll be looking at how they may be associated with things like telomere length, different inflammation and steroid profiles, and many different outcome measurements. And this is just some, data from the microplastics methods, showing that we can essentially measure a range of polymers in placentas. And we're finding that, the majority of placentas we're characterizing essentially have at least one microplastic detectable in it, if not more. But we're also trying to develop, really foundational studies that tell us more about both this in utero exposure as well as maternal transfer of microplastics. So, in collaboration with our team at USALT University in Belgium, they're doing de novo sample collection that will enable us to look across maternal blood and urine samples as well as cord blood and placenta measurements to see how the maternal levels of microplastics influence the in utero exposure to these compounds. And with collaborators at previously at the University of Colorado, now at Johns Hopkins, we're also trying to establish cohorts that allow us to evaluate the impacts of microplastic exposure in early life. Unfortunately, when we are collecting samples for microplastic measurements, we have very strict procedures we need to follow to avoid contamination of the samples. So we don't have compelling evidence yet that biobank samples are necessarily useful for microplastics analysis, although we are hoping to demonstrate that in the near future. But it's very important to start thinking about how we can design cohort and prospective studies to do suitable collections for measuring nanoplastics and microplastics to see how they're associated with different developmental outcomes. So just in closing, some of our ongoing areas of development for advancing micro and nanoplastic measurements in different tissues. The goal is to really have optimized methods for multiple biological specimens, whether that's blood, breast milk, stool, reproductive fluids like follicular fluid or seminal fluid or lymph nodes. Essentially, we're trying to have methods for many different biological specimens. But at the same time, we also want to increase the scale that we can do this and increase our throughput. We're purchasing more instrumentation. We're trying to think of ways we can make the sample preparation more efficient so we can essentially analyze more samples in the same amount of time, as well as detect more polymers in those samples. We're also trying to identify some microscopy approaches we can use to kind of validate some of these mass-based measurements. And we're very thankful to have different resources at the cores here at Emory to be able to do that. But really, our hope is to continue the development and application of these methods in both general populations as well as clinical cohorts to really assess the impacts of micro and nanoplastic exposures. So in conclusion, plastics, represent a very complex exposure burden in humans, and the way we're currently characterizing these does not account for that complexity. So we're very interested and focused on leveraging these untargeted high-resolution mass spectrometry methods to really expand our plastic exposure assessment and better embrace this concept of the plastic exposome. And we're leveraging these approaches in foundational studies to really try and estimate what are the current exposures to microplastics and how may those be impacting different health outcomes. So in closing, I'd just like to, acknowledge different funding sources that have supported this work. If you're interested in learning more about the Aurora project, you can scan the QR code. And of course, I'd like to thank my, team who has really contributed to many of the different aspects of this work you're seeing. They've just been, excellent to work with and really enjoyed um, my time here at Emory. Thank you. I've got about 4 minutes for questions, so happy to take those.
Thank you, Dr. Walker. That was incredible. I think many of us I have obviously heard of microplastics, but are not familiar with just the level of complexity of their analysis, as well as the range of impacts that y'all are already discovering in that field. Before I open to questions, I want to just sort of make one quick announcement, acknowledging that some folks had a difficult time accessing today's event because there was a Teams invite that came up. So just for folks to know, Teams will always be a glitch. We are not meeting on Teams for DOM Grand Rounds. And there will be grand rounds next week. We are currently working with our our DOM sort of communication team to troubleshoot that on updating the calendar, but please do join us next week. And with that, I will open the floor for questions.
Thank you, Dr. Walker, for this nice talk. I wanted to know if this method can be adapted for analyzing the microplastic in mice samples, like in mice blood plasma.
Yeah, absolutely. And I think those are very important samples to work with and very important studies to start thinking about establishing. So there's no reason that they couldn't be extended to different experimental systems. It would just be good to discuss a sampling strategy so we know that the samples are being collected in a way that minimizes plastic contamination. So for example, if we're analyzing blood samples, we actually prefer whole blood because if you're going to spin out the red blood cells, you'll actually influence that
Mhm.
distribution of the particles in the blood. And so we prefer the blood just be collected in a glass tube and then just stored as is.
Okay, thank you so much.
What other questions y'all have?
Hey, Dr. Walker, thank you so much for this discussion. I think my question is really because as a practicing clinician who is very concerned about micro and nanoplastics and their ultimate effect on disease, I think this has been so fascinating just to hear about how intricate this process is. As a matter of fact, and this is just a comment, as a matter of fact, when you were talking about like, well, you think about all the supplies you have in the lab, and how do you know that you're not going to be contaminating your work with all of the plastic that you're using to do the work? So when we're thinking about, patients often ask me about like, are microplastics a problem? Did microplastics cause my disease? It sounds like we're really, with the cutting edge work that you are doing, it does sound like we're still quite a ways off from being able to really design that level of a study.
Yeah, absolutely. And I think it's very important to also consider, kind of the risk benefit of plastics, right? So, we talk a lot about, considering plastics exposures, how those may impact our health, but, there's also a lot of benefit to them, right? So like with the patients you're treating or the interventions you're doing, plastics are necessary. And so I also, I feel a little hesitant to say that, you know, we should necessarily be looking for disease relationships now. I think what I'm mostly interested in and is just understanding these exposures and also trying to develop the evidence we need to, see how we should be designing studies to study health outcomes. I mean, I can tell you I get attacked. I don't know if attacked is the right word, but spammed by comments and posts on LinkedIn whenever I post anything about microplastics saying there's no evidence these are bad. And then they'll post a bunch of links and then you'll click the links and it will just go to blog posts by the same person or whatever. But there's definitely a PR and industry push to kind of minimize some of this research on plastics and whether these microplastics are harmful. And so I think what I really hope with our methods and science going forward is that we can just communicate the data in a transparent and intelligent way where we can, have accurate discussions about this risk benefit relationship.
That's very helpful and so important in terms of just the ability to access that messaging for so many folks out in the general population. We do have a question in the chat. It appears we are only at the point of measuring permanent exposures that are found in tissue. And what about transient exposure effects? And as an endocrinologist, the important effects of the associated chemicals such as endocrine disruptors.
Yeah, so that's a great question. So, the one of the challenges and complexities we face in exposome research is that, unlike your genome, which is relatively static your whole life, your exposome changes daily, hourly, by the minute. My exposome at my house is very different than in my car or in my lab. Hopefully, it's better in my lab than not in my lab, but. And so how do we address that transient nature of the exposome? And that actually is kind of you know, a good way to go back to our kind of other research focus, which is developing these higher throughput exposome methods. Our goal with those is to actually have very high throughput and cost, effective measurements where it may be possible to do repeat measures or to look at somebody's endocrine levels, across multiple or endocrine disrupting chemical levels across multiple time points. So we're not just trying to do a single measurement and infer information about an exposure that may only occur at a week ago and then be gone. And so, I kind of see that as a scaling. The way to get at that is through scaling and make these methods more suitable for application on a larger scale and, to do them in a more repeatable way. But yeah, that's definitely an important consideration. The other thing to kind of recognize is that for a lot of these endocrine disrupting exposures, especially from more common commercial products, is they're not episodic, they tend to be more chronic. And so we may actually develop kind of a baseline exposure level of many of these, which we can measure, at different times and kind of use that to estimate some type of exposure assessment.
All right. Well, with that, we are at time. Thank you again, Dr. Walker, for this incredible presentation. Thanks everyone for being here. We will see you next week for Department of Medicine Grand Rounds. We'll have updates in infertility. Thanks y'all for being here. Have a great one.
Thank you.